it's just for everyone, I think, because we do hear about some of the confusion that are there with all the resources that have been shared. So I just wanted all of us to go through some of these resources and to understand where to find the things that we discussed today. So this is the pre-registration page where we all started, and like many of you have pre-registered here. So in this page, we have a video overview, and then we have the session details. Along with that, there's a form that many of you have filled up, and then we send out the emails. So when you go through this page and you would want to learn more about the sessions, so the second thing that I would like to take you to is the program syllabus and how we have planned the live sessions and the resources that are associated with it. So one of the exciting part of this program is just which also differentiates us from other similar online programs, is that there are several resources that are associated with each of the programs. So this is the syllabus page, and Sonalika, if you can paste the link of this page too, so that we can all
Yes.
browse through this page. So as we go to this page actually describes the curriculum that we have developed, and also the resources that would be associated, including the assignments, the hands-on sessions, and what will be covered in the hands-on sessions, and where would you learn about those technologies, and what all Ilya just said about getting started with big data, getting started with gene expression data, getting started with um, genomic data, and then applying it for cancer problem and applying it for infectious diseases. So there's a lot to learn, and there is, so I mean, I'm really happy that we have all of you together today to showcase some of these that we have put together for this program. So there's a video again which gives an overview of what was covered. So you have already seen what was covered. So session wise, when we go to when we will start, which would be on Tuesday. So we are going to start next Tuesday at 9:30 a.m. CST. I think it's 10 a.m. CST. So yeah. So in the first session, we will be covering about bioinformatics and translational oncology research. So this would be the overview of the cancer problem. As we were discussing, we will discuss about the complexity in the sense of how it is being described in at tissue, organ, and cellular level, and then the molecular factors that actually are associated with tumor development, growth, and metastasis. Then we will understand that the how genotypic and phenotypic relationship can be identified through these clinical and molecular data types, and then we will go through the overview of the platform and what we will be doing throughout the course. So this will be on the first session, which will also be available to all of you. And the associated online resources, as I was telling you, that at we started off with the learn site. So let me go to the learn site. This is the associated courses that are available on the learn site. So if you have not created a login for yourself, so please do that. This is the link, and let me show you how you can do that. It's very simple, and this is also also for those who would not who are participants, who would be participants, and also for those who would think about joining later. But it's great to have you here. It's a free login, so you can just log in with your email address or your social media address. So I'll just sign in. So once I sign in, I'll come to the home page. I'll just show you how to navigate through this website. So once you come to the home page, it describes all of what we described and discussed today, as well as what are different specialization that you can opt for. So we have specializations in oncology, infectious disease, neuroscience, agriculture, and then the T-Bioinfo server, and also all of the things that we discussed, our associations. And then the interesting thing about the syllabus is that we will start with introduction to bioinformatics. So let's go to our first course, introduction to bioinformatics, which will be covered in the first session. So in the first session, we would all want to learn about where bioinformatics can be applied, especially in biomedical research. So all the areas that you might be interested in, you will be able to navigate through this. And this course also has a form which let us know more about you, about your research interest. So this is a course that I recommend you all to take, and it's a free course. So once you create your login here, your free login here, you can start taking this course after your login is done and you have a profile. So let me quickly show you what a profile looks like. So this is my academic profile. I have a image, and I've described myself. So one of the benefits of having a profile is that you will be able to see your progress, how many courses you have finished, how many programs that you are enrolled in, and also like what are the, as I said, that what are the courses that I'm enrolled in, I'm doing, and what are the programs I'm enrolled in and I'm like interested in going through, and then the certifications that I've gained from taking the courses, these courses, or completing the programs. So all of this you will gain and get from this profile once you just create a free profile here and put some of the information that would help us learn more about you, and also also people who are interested to collaborate with you, or people who are interested to hire you for any specific research work. So all of that is actually are the components of having the profile. So this I would really recommend you to all of you to create and at least go through the free resources that are available with all of the sessions. So as I said, that the first session that would be about introduction to bioinformatics and introduction to cancer. The second session is more technical, where we dive into genomics. So in genomics, we will learn about next generation sequencing. We will learn about how we can analyze genomic data and find out those variants and those variations and those mutations, significant mutations. So again, there is a bunch of courses that explains genomics, and one of the benefits of doing this is not just that you would be able to learn from the very beginning, from the very basic, starting from the DNA structure, but also you will have an option to work and learn R and apply R or Python using our coding playground tutorials. So here I will show you another demonstration where you will able to also apply data science techniques. So about genomics, you will be also be able to go through the hands-on sessions, and the hands-on sessions with the star mark are the ones that are available to the participants, and also to those who would want to subscribe for the asynchronous material. So when I click into this, I'll get information about how I can prepare my pipeline and understand about the process of creating a pipeline and what are the components that are involved. So once you complete, this is like an achievement that I've gone through this tutorial, and I have gone through this tutorial. So, but not yet on the platform. So here you will also learn about the data sets that are embedded in the with the link that you will get. So these are all the benefits of getting to the asynchronous material and going through the tutorials, and that goes from basic introduction to building up variant pipelines to do a phylogenetic analysis, and again go through multiple sequence alignment to see the variation and evolution of genomes. So obviously there are so many applications which I cannot list out in this session. However, the second session and the third session after the second session learning about genomics would be about transcriptomics data, so gene expression data. Here also we will go through an overview of how what is the significance, what is RNA sequencing, why it is done, its history, and then about its application, that how do you do it, and get understanding about the quality of the file that are available, and then how do you pre-process the data, map it to the genome, and going through all that process. So we will be doing this on the T-Bioinfo platform. So just let me quickly go to the T-Bioinfo platform. So sorry about this. This is a different pipeline. So this is the TBA Info platform, which we discussed in this meeting, and which is a really amazing platform, which is a very comprehensive as well, describes all of the NGS data types, and shows us very elaborated methods and pipelines to analyze this sort of data. So this is a typical pipeline. This is how it looks, and one of the best part of this is that it's the entire platform is very friendly, because it describes all of the processes, all of the data types that are there, and what are the applications that we can do with this. So when we go into this section, this is about transcriptomics, and I was discussing session about the transcriptomics. So here we will be going through the example data sets that we have compiled for you and learn from. So this is a pipeline which has already inbuilt data sets which are put together, and you will be learning about those data sets in the session. And then we will be going through the pipeline together, building a pipeline, how this format makes sense, why these are placed in this manner. So the flow is that these algorithms are placed in, I know many of you are already aware of this, but for those who are not, are in a logical manner, put together in a logical manner, where we start from pre-processing the data to then error correction, then maybe mapping it to the genome, or doing isoform construction, or doing mapping it to the transcriptome, or doing differential gene expression. So there are several experiments. I mean, you can build from different paths that you can take to analyze your data. So in this session, we will be learning about the breast cancer sample cell lines that we discussed, and the publication that is associated with the session. So I think I can go about it a little bit more, but I think we don't have that much time. But coming to the part where we will be discussing about the application, and there was a question about classification and clustering in the chat. I think it is something that also I would love to talk more and more about, but we already have a session described for this, where we will be going through machine learning. And in this, we will be applying gene expression data and finding patterns from the gene expression data. So in this process, we will be applying machine learning methods using the TBA server, obviously. So we have those very well defined in these sections of data mining. So you will be able to do clustering and PCA, and also if network. But yeah, you can start with all of these methods when you start with the program. And also, like, to those who are joining, I think it's great to start with the coursework and get a glimpse of the sessions before actually the start of the program. So now would also want to tell you more about a little bit of machine learning in biomedical data and data science. So what is the data science aspect? So sorry, let's go back to the courses. And let me search for data science. So we have a lot of data science courses, and one of the example I would want to show you is that Introduction to Data Science for R. So here we have a lot of tutorials that you will be able to go through, and it's relevant to the session. So we will be going through some of the visualization techniques. How would we do PCA and using R code? So as I said that R Studio is available here on the browser, so you can actually learn about the problem here, and then learn about the data that is available, and then the commands where it is described what it explains, like what data set that we are taking, what kind of step that we are doing. So here, once you learn about these steps and learn about what will be done with the data, you will have an opportunity to do it yourself. So here's a big tutorial. So learning about all these techniques, going through the assignments, and you'll be able to run it yourself. So this is a code block where this you can run the scripts by learning about the scripts and then actually doing it yourself and getting results. So for the data sets that maybe our researchers are working on, our research fellows are working on, they're utilizing these tools to come up with images and for presentations and publications, etc. So this was about that. And then the fifth session is about oncology, where we will be learning about the oncology data sources. So it's more about the data repositories. So we will be going to NCBI. We will be looking at the data sets that are available there, learning from them, taking some of them as examples and understanding how they connect. I mean, how do you annotate your results better? So how do you annotate your mutations? How do you annotate what in information that we are getting out of the machine learning after the machine learning analysis? So this is the first phase. After the first phase, second phase is all about infectious disease problems. In this, we will be learning about viral genomes and host transcriptomes. So we have also virology sections that will be utilized in this program. So going back to that, so here we will have examples projects in this time. From this part, you will start working on projects. You will have an opportunity to start working on projects. So these projects are asynchronous projects that you can take anytime, and you will have an opportunity to work them through when you have availability. And these are like very descriptive projects that describes yeah, I mean, these are some of the data sets that are described by publications, and we have put together how do you analyze and understand from what the authors have described. So these are some of the example projects, and then we will learn about genomic multiple sequence alignment and phylogeny. And we have another few projects like COVID-19 and Ebola. So these are again about the mutations and how COVID-19, or I mean, this is from a paper that describes the proximal origin of SARS-CoV-2. It was published in Nature Medicine 2020. So yeah, you will learn about this, you can learn from this project as well along with the session. So these are the live sessions that we will be covering, and along with that there are always asynchronous material available for you to take throughout the week. And then we will go through project examples where we will learn how to compare and I mean, these are different biological questions that we are asking from the data, so that you get an experience of how can you ask your question from your research. So again, like after learning about all of this, the last part of the session, the 10th session, is about host pathogen interaction. So here we will learn and connect some of the aspects of our omics data to structure, and finding those key mutations and understanding those, and then the effect what it caused for the function of the protein. So this is what is planned for this precision medicine program. And I hope that I've been able to describe this well to all of you. And I'll be happy to answer any questions that you have.
And also, I wanted to add that signing up on the Learn portal, that is learn.omicslogic.com, would get you the automatic access of the T-Info server as well, which Dr. Mohit just shared. And yes, so we'll also share, I would be mailing you all the details which were discussed in this session, and also would give you the links and how you can proceed further with your submission.
Right. So thank you, Sonalika. I just wanted to add a couple of things. So we also have a Slack channel, and I forgot to mention about the program part. So yeah, when you go to learn and when you do this login, you'll be able to go to this tab programs. So in programs, we have all the programs listed out that are upcoming or have been ongoing. So you will find precision medicine here. And to register for this program with live sessions and everything, not just the asynchronous material, you have to start your application. So using this link, And once you start your application, you can select different options. So these options involve all the cloud access. So there are 5 terabyte of space that would be allocated, and you will be able to run the cloud pipelines, and then the coding sessions. All of this is included in this package. And here you will be also able to apply some of the discount codes or some of the fee waivers that have been provided by Sonalika. And also for lower income countries, we have a scholarship option that is also available. And I would like to request those who are applying for scholarship to do it as soon as possible, because we are going to start the program on Tuesday, so we can process your application by Monday. So I would also request Sonalika to share the scholarship link. So there are different access levels, so different licenses. Another license is about the intermediate step where you would want to have an additional one-on-one mentor support, where you would want to go through some of the materials along with the mentor, ask your research question. So even in this entire program, and even in the webinars that we do, it would be really nice to discuss your research question. But yeah, obviously there's a limitation of time. But when you are in one of the programs, then it would give us like a lot of time for you to discuss your research and work on your idea, and if we can utilize these resources to be able to come up with the research outcome. So the research part is also being in this advanced license, so it gets you to work on your own project and additional month of cloud access. So this is the advanced license. And then obviously there you will have access to a lot of courses. And then once you go into the sessions and like register for the program, you will be able to see a different interface. So I'll take you there. But you will also be able to come up and it will also be able to get your certificate, as I showed you earlier. So this is the registration part. And along with this, now let me show you. I'm not registered for this program, so I would just want to show you another program and the interface. So once you're registered, this is the interface that you get. So you will get the courses that you're doing and all of that, and you can ask for a Q&A, or you can ask for a mentor session, and then you'll get your certificates here. And when the sessions are ongoing, you will find the session videos here. So the video recording, you will get into when you get registered, and then you will have access to an interface like this. And then you will be able to get the session recordings into this interface, so that you wouldn't have to go anywhere. It's the same place where you will find all the details. So also the same thing for the Zoom link. For the next session, you will be able to find the Zoom link as well. So this is another program that is ongoing for our partners in Louisiana. So it's a program with LBRN network. So I think, yeah, that's it that I wanted